Thank you for the introduction. I'm going to be presenting methylation sequencing with Oxford Nanopore Technologies. Just a brief overview before we get into the content with Tim and Jeffrey. I have 5 topics that I'm going to cover: introduction to nanopore sequencing technology, introduction to methylation, the different methods for doing methylation detection, how methylation works with nanopore sequencing, and why you should choose nanopore for methylation. Nanopore sequencing has 4 key components. On the left-hand screen here, you see the nanopore, which is in blue, the motor enzyme in purple, the DNA or RNA that's flowing through the nanopore with the help of the motor protein, and then the supporting membrane that houses the nanopore. On the right-hand side, you can see as the DNA is flowing through the pore at about 400 base pairs per second, it's turning the sequence of the DNA into an electrical signal that we can detect. And in the case of methylation, it is determining a different methylation signature versus a canonical base. And what this means is that we're directly sequencing DNA and RNA, and that means that you get richer information, specifically these modifications like methylation. This is done without PCR, so there's no amplification bias. It's read length agnostic. You can go 20 base pair reads to 4 million base pair reads. Analysis is done in real time. This technology is amenable to many different formats, so it's scalable both for low throughput applications as well as high throughput applications. We deliver the nanopores on 2 different flow cells, the MinION flow cell and the Promethean flow cell. The MinION flow cell has 512 channels and does about 15 to 35 gigs of genomic DNA output at 25 base pair reads, or ultra long reads, which are 50,000 base pairs or higher, and does about 10 to 20 gigs of output on that flow cell. Again, any read length, and you can wash and reuse the flow cell. The Promethean is about 5 times the size of the MinION with 2,600 channels and does over 100 gigs of output in the traditional 25 gig long read format. The MinION has 2 different devices that it runs on. The MinION device, which is a single flow cell device that has to be connected to your own computer for data processing. And then our larger MinION device is the GridION that has 5 flow cell positions. Those flow cell positions, like all our devices, are independent of one another, so you can start and stop them, run different assays on different flow cells. The GridION comes with onboard computation, so all the base calling alignment can be done on the instrument. The Promethean flow cell has 3 instruments. The Promethean or P2 Solo, which is the one here on the left, has no onboard compute, has 2 flow cell positions. The P2 integrated in the middle here has onboard computation, 2 flow cell positions. Again, individually addressable flow cell positions. And then our high throughput instrument is the P24 on the right here with onboard compute and 24 individual flow cell positions. Again, totally independent of one another, so you can sequence 24 different assays at different times on that instrument. Very quickly, I'll go through just some brief introduction to methylation. So methylation can occur at both adenosine and cytosine, but the one that we're most commonly looking at is cytosine. And the most common methylation is the attachment of a methyl group at the 5 position, and that's called 5-methylcytosine, or 5-MC is what most people will call it. And then a more rare modification is 5-HMC, that's 5-hydroxymethylcytosine. You'll hear about CpG sites. CpG sites are cytosines followed by a guanine. The P is for the phosphate between those two, and methylation most commonly occurs at these CpG sites. It's a genomic region that's usually 200 to 2,000 base pairs. 60 to 70% of gene promoters are associated with CpG islands, and CpG islands often overlap transcription start sites. So it's really critical for gene expression and regulating gene expression. And there's 40 to 50,000 CpG islands in the human genome. Methylation, because of that role in regulating gene expression, methylation is involved in embryonic development, genomic imprinting, X chromosome inactivation, stem cell differentiation, silencing of different genomic elements, chromosome stability, and aging. Methylation sequencing can also be used for haplotype phasing. It can be used for chromosome parent of origin determination, as well as tissue of origin determination, which is what we're going to learn more about today. There's two methods that have been used conventionally for methylation detection, short read sequencing, and then methylation arrays. With both of these methods, you have to convert using bisulfite, convert the cytosines to uracil. The unmethylated cytosines get converted to uracil, and then they appear as thymine, and the methylated cytosines are protected, and they remain as cytosines. This is a fairly harsh treatment that damages the DNA. In short read sequencing, you lose strand complementarity. You can't distinguish 5-MC from 5-HMC. The process is fairly laborious. It's subject to PCR bias, and it's short fragments, so you are unable to phase large regions. On the array side, similar challenges with DNA damage. It has really limited content, so you can only see up to 3% of CpG sites. You can't distinguish 5-MC from HMC. You get some probe reactivity that will hybridize multiple loci. You can't detect the non-CpG methylation, and there's no sequence context. With Oxford Nanopore Technologies, we have the fastest workflow for methylation sequencing. And the reason it's so nice is it's part of our traditional DNA workflow. It's not separate from our DNA workflow. So the only thing that you have to do to enable methylation sequencing on our platform is the same workflow that you were doing to produce DNA libraries for sequencing. And then in the software, you simply click a button that says turn on methylation sequencing, give me that data. So you start with DNA, you do a quick end repair and cleanup, ligate or adapters, that motor protein I showed you earlier, clean it up, QC it, load the flow cell and sequence. So you're from DNA to sequencer in less than an hour. And sequencing is really fast because we're doing the analysis in real time. And then this flows into a full suite of analysis tools that allow you to look at every methylation site on every cytosine across the entire genome, or you can down select from that. So you can only look at CpG sites, or you can only look at CpG islands, and that's all up to you. You can also do a targeted approach, which does not require any additional library prep. You just do on flow cell targeting that allows you to select which CpG islands you'll look at so that you can get more samples on the flow cell. Lastly, the benefits of doing methylation sequencing with Oxford Nanopore Technology is it's simple. The methylation is captured as part of our DNA sequencing workflow, and you don't have to do any conversion step to get the native DNA with methylation is what we sequence. And that makes this workflow really simple. Also is context. We can distinguish 5-MC from 5-HMC. We can phase at nucleotide level precision. We can profile methylation in challenging regions like repeat regions, and we can capture methylation in CpG and non-CpG contexts. And then it's a very flexible platform. You can capture 100% of methylation sites, every cytosine, all contexts, or just CpG or just CpG islands, and you can target specific sites without any additional lab work using our adaptive sampling technology. To get started, visit our website and go to our methylation page. We have tons of content, videos, getting started guides, and publications and posters that will help you get started with methylation sequencing on Oxford Nanopore Technologies. With that, I'll turn it over to Tim to go into more detail about some of these specific applications.
All right, thanks, Alex. Excited for the opportunity to talk about Wasatch Biolabs. I'm one of the co-founders for Wasatch Biolabs and Renew, and we've worked really hard to try to leverage Oxford Nanopore Technologies in our lab, specifically to utilize it for methylation applications that are novel for both research and pointing towards clinical utility in the future. Our lab, just in by way of introduction, is really focused on innovation and utilizing new technologies that can facilitate better epigenetic data. My background is in epigenetics. I've studied specifically DNA methylation my whole career, and focused highly on new and novel ways of discovering methylation patterns that can be predictive of outcomes for both research utility and, again, eventually pointing towards uses that are more pronounced and profound in clinical settings down the line. Our lab focuses on Oxford Nanopore sequencing. We specialize in it, and I think have gotten quite good at utilizing all of its potential in methylation sequencing in particular. One of the reasons that we focused on Oxford Nanopore technology, it was really nicely outlined by Alex earlier, is that we get more information than just the sequence itself. Our focus is definitely on leveraging both genomic data and epigenetic data. And really, at the heart of what we do, we want to focus on taking the DNA for what it is. We don't want to modify it. We don't want to amplify it. So we wanted a technology that enabled kind of size agnostic sequencing, like it doesn't matter exactly what size of molecule we're starting with. We wanted to be able to sequence whatever we had available to us without conversion, either by sulfite conversion, as was mentioned before, or even enzymatic conversion, and certainly without amplification. We want to see what is natively available in the DNA, as that helps us to more accurately point to what issues may be happening in the DNA without technical bias, which should eventually give us the best quality data and thus the best quality predictors in the future as we move forward. I'll introduce you to one use case of how we kind of go about our business at Wasatch Biolabs. This is one that we've developed internally, but we also focus heavily on working with other groups that have diseases that they're interested in, or specific questions where we can apply epigenetic data or genomic data that's available from Oxford Nanopore sequencing to their specific use case. For us, I'll introduce you to this one assay that we call the NeuroLens assay. This was developed by myself and the co-founder for Wasatch Biolabs, actually. His name's Chad Pollard, who was a graduate student at the time with me. I've leveraged DNA methylation analyses in my own lab for many years and have focused on tissue of origin prediction using methylation for that purpose. For many, many years, Chad came into my lab and looked at some of the tests that we were doing and said we could do this for Alzheimer's and for neurodegenerative disease if we just looked in cell-free DNA. And I said, go explore it. And he did, and found some incredible things in that effort. So really the problem that we recognized was that neurons can die without our knowledge, and can do so quite frequently, especially later on in life. In fact, most of the diseases that we know as neurodegenerative diseases start long before we ever recognize any symptoms. And this is problematic because we don't have a way to track those. And so we needed a technique that could potentially identify those signals of neurodegeneration much earlier than was currently available for us. And not only that, we wanted to be able to detect not only neurodegeneration, but what regions of the brain or what cell types might be dying, because those are indicative largely of the disease type itself. For Parkinson's disease, you have loss of dopaminergic neurons. For Alzheimer's, it's loss of cortical neurons and so forth. So if we could produce an assay that enabled us to not only detect early the fact that neurodegeneration is occurring, but also enable us to discriminate between different cell types, you could imagine that we could use these to track therapeutic response. We could even identify better patient populations for clinical trials, for example. We could detect them early and determine who has specific phenotypes that might work best under certain conditions. So we were excited about the prospect of pursuing that question, and we found some pretty incredible some pretty incredible things as we've gone through this process. The premise of this, as was mentioned before, is all focused on DNA methylation. DNA methylation literally acts like a fingerprint for individual cell types. And so if it is true that when cells die, they release their contents into neighboring tissues, in this case, neurons die, they release their DNA and other contents eventually into the blood where it gets degraded, then we have this small snapshot if we look at the cell-free DNA fraction of blood plasma of what cell types are dying in the body. If we can use methylation then to identify where that cell originated, then we can determine what percentage, what fraction of the cell-free DNA in the blood is coming from different cell types. And that's precisely what this assay is designed to do. We started by very conventional means. This was done in 2020 and 2021, most of this work, where we leveraged methylation array, as was mentioned earlier, as a common discovery tool. This requires bisulfite conversion. And we utilized the array to identify positions that discriminated between cortical neurons and blood plasma. And you can see here on the left that we found a number of those positions, about 10,000 or close to 10,000 positions that were different between the blood plasma background and cortical neurons. Now we whittled that down and we performed bisulfite conversion and PCR amplification of a handful of sites that we could then sequence. In fact, in this graph here to your right, this only used one position in the genome where we knew it discriminated the methylation signature on that position or at that position discriminates between cortical neurons and blood plasma. You can see that on our y-axis here is the fraction of neuron-derived DNA. And if we look at just on the far right-hand side, just at cortical neurons, you can see that the vast majority of that is coming is neuronally derived, right? It's coming from a cortical neuron. If you look on the left-hand side at this one position, patients with active Alzheimer's had high levels of neuron-derived cell-free DNA in their blood plasma. Young controls had virtually none. If we skip over here to mild cognitive impairment that eventually went on to get Alzheimer's, they also had elevated levels of neuron-derived cell-free DNA. And patients with mild cognitive impairment who did not, within 5 years, go on to get Alzheimer's most had no elevation in neuron-derived cell-free DNA, with a few that did have a small amount. The most interesting category here, though, was that age-matched controls, about 90% of them had very little, I should say, neuron-derived cell-free DNA, but about 10% of them had elevated levels. Now that could either be, in this case, a false positive, or this actually matches very well. Individuals over 60, about 9%, 9 to 10%, will eventually succumb to neurodegenerative disease. And so we believe this actually represents a pre-symptomatic detection of this neurodegenerative disease, though we can't confirm that in this specific patient population, but it's intriguing. Now, this initial work was very exciting for us, and it demonstrated that this idea of using DNA methylation for tissue of origin detection could have utility in neurodegenerative disease. But there was a number of things that we had to overcome for us to be able to do this the right way. And we quickly realized that this idea of bisulfite conversion, PCR amplification, created too much noise for us to be able to accomplish our goal. And so we shifted to Oxford Nanopore Technology, and I'll show you some reasons why. One of the biggest reasons was when we looked at array data specifically, which utilizes bisulfite conversion. It was our tool for discovery, but it was on the table for us to potentially use it also as a tool for using as an actual test. One of the issues that we kept finding, though, was that there was a large degree of batch variance with array. So if you look at this principal component, the first principal component, the thing with the most variance, was actually the difference between location 1 and location 2 of the same samples. So we have this issue of batch effect that's going to be very difficult for us to parse out in the future. We don't have that same issue with ONT looking at these same samples. But we also wanted to know, because kind of the gold standard at the time has been array for discovery studies. We wanted to see if by using ONT for our discovery, if we would miss much that we would have identified on array. And we found that we identified approximately 90% of a little over 90% of what was found on array using ONT with low pass sequencing. About 5x coverage is all we needed. But in addition to that 95%, we found far, far more positions using Oxford Nanopore than we did with array. To the tune that this was originally done just between two different tissues to show you how different these results could be. But we can really highlight that when we actually built models or did analyses specifically for cortical neurons, spinal motor neurons, and dopaminergic neurons. If we used the array, we found thousands of positions. If we used ONT in our lab, we deem this as direct methylation sequencing. We found millions of regions or positions in the genome that were actually discriminatory and that we could use in our models. And so we used ONT as our platform for kind of discovering these positions. And we assessed multiple different cell types to ultimately construct models from which we could use as predictive tools for, the fraction of cell-free DNA that's coming from different types of neurons. So this figure is one that's got a lot of information in it, but effectively what we're seeing is on this first bar, we're looking at cortical neuron DNA methylation profiles versus pretty much everything else. And so you can see the total amount of sites that discriminate just cortical neurons or just spinal motor neurons. And I'll skip over here. We can also see just dopaminergic neurons. One of the problems that we kept running into too is even though we might be able to discriminate between Alzheimer's and control individuals, it became very difficult to discriminate between somebody that had Alzheimer's and someone that had Parkinson's or ALS or something along those lines, because there's a lot of what we would call pan-neuronal DNA methylation signals, meaning signals that were true for pretty much all neurons. And so we needed some capacity to discriminate between, not just between patient and control, but also between patient and other types of disease. So we leveraged this idea that if we had models specific to cortical neurons or specific to spinal motor neurons, perhaps we could then leverage that for better categorization of patients. So we did just that. We built models. We built models that specifically detected cortical neurons, dopaminergic neurons, spinal motor neurons, and so on, and even some glia. This graph here is just showing that this has potential to work. This is computationally derived models that are looking at spiking in reads from known cortical neuron DNA into blood plasma as a background in known amounts. And so we're effectively looking at our capacity to take random reads taken from cortical neurons, spiked into blood plasma, and being able to predict how much we spiked in initially. And you can see that when we did this with our cortical neuron model, it worked beautifully. Same with dopaminergic. Spinal motor is the weakest model that we have right now. We're working on improving that, but even that showed some promise. And we took this and then said, okay, it works in theory in our own data sets, but does it work in patient populations? And if so, how can we leverage that for anything moving forward? We took those same models and we assessed their performance in different disease states. So if you look at this top row, starting from A, B, and C, we looked at cortical neuron models, dopaminergic models, and spinal motor neuron models. If we look at the cortical models, we would expect to see, because Alzheimer's is a disease that is mostly associated with cortical neuron cell death. If we're looking at at the percent of cortical classified reads in blood plasma, cell-free DNA from blood plasma, from these individuals with Alzheimer's disease, mild cognitive impairment, and others, we'd hope that the highest level is seen just in Alzheimer's disease and mild cognitive impairment, which can tend to move towards Alzheimer's disease. And sure enough, that's exactly what we saw. You can see Alzheimer's disease and mild cognitive impairment has great separation from control on its own, but also in this case has pretty good separation from other disease states, other neurodegenerative disease states, Parkinson's disease, familial and sporadic ALS. When we do the same thing for dopaminergic neurons, the hope is that we'd see separation with Parkinson's disease or elevation with Parkinson's disease. And we do get great separation or great kind of higher enrichment of dopaminergic classified reads in Parkinson's disease, especially when compared to controls. But you can see an elevation of these other disease types. So here again highlights the problem. We want to be able to say definitively that something is Parkinson's disease and not just general neurodegeneration. And you can see how difficult that could be. There are lines that we could draw here to give some confidence, but it's not as much separation as we would like, despite the fact that we are getting some separation. Spinal motor neuron model, you can see the same thing. ALS both increases, especially compared to control. But when you compare it to other disease states, it's a little bit harder to parse out. So we took all this information together and said, well, what if our classifier was not just one specific type of neuron, but we looked at all the different classifications of neurons. The simplest form of that can be seen in panel D, where we looked at two distinct classifications, dopaminergic and cortical. When we look at dopaminergic and cortical and we look at age-matched controls, they're all in the lower left-hand quadrant here, which is what we would expect. Parkinson's disease separates nicely and has elevated dopaminergic neuron DNA, and Alzheimer's and mild cognitive impairment separate nicely and cluster on their own, having high levels of cortical neuron cell-free DNA present. So you could take that same idea and even try to plug in a different disease state like ALS. When we did that, still just classifying dopaminergic and cortical neurons, again, those are not the targets for ALS that we would want, but this was trying to discern whether or not this would muddy the waters. And you can see that it kind of does. If we look at sporadic ALS and familial ALS, which are pink and green, you can see them occurring here kind of in the middle. They do cluster, but they cause a little bit of noise. You still get the separation of Alzheimer's and a little bit of Parkinson's and controls, but this addition makes this more difficult. Visually, it's harder to show things in more than 2 dimensions, but we can generate models that do this. And we did actually generate a 3-dimensional version of this where we have dopaminergic, spinal motor neurons, and cortical neurons all being used to try to separate out, to parse out these different disease states. And you can see really, it does this really nicely, right? So we have our control patients here at the bottom, our sporadic ALS and familial ALS here on the right-hand side. Dopaminergic cells indicate Parkinson's, high levels of dopaminergic cell-free DNA indicate Parkinson's disease here at the top. And we have mild cognitive impairment and Alzheimer's over here on the left. We can build models that do exactly this. It becomes difficult to visualize once you get over 2 or 3 dimensions, but we can build models that utilize all of this quite easily. And so this is depicting only Alzheimer's and mild cognitive impairment patients on these slides, on these rock curves, but you can see how powerful this can be. Alzheimer's too, I should note, or cortical neurons, I should note, were by far the best model that we had. This pattern that I'll show you right now only becomes more pronounced when we look at things like Parkinson's disease and ALS. But if we're looking at Alzheimer's disease, if we look at Alzheimer's disease versus control, the cortical model identifies it very nicely. Even the dopaminergic and spinal motor neurons identify it nicely because there are pan-neuronal signals. But the combined model always performs as good as anything else, better than anything else. The problem, though, had always been, can we do this same thing not against control, but against other diseases as well? And so we performed that same test. You can see that quickly starts to fail in our dopaminergic models and spinal motor neuron models, but the combined model always performs quite nicely. And we can see this even again more pronounced when we look at Parkinson's disease or ALS. So we've gotten to the point now where we can discriminate between these different cell types, and they provide a lot of extra information that enables us to discriminate between different disease states, all different forms of neurodegenerative disease. So what are we doing right now with these data? We are currently working with a couple biopharma groups to assess therapeutic response monitoring. We're in the process also of beginning to build an LDT validation work for this test. Um, from there, and we've already begun this process too, we're looking at early identification studies. The hope is that not only are we able to determine that somebody has Alzheimer's or Parkinson's, but that we're able to do it well before symptoms ever onset. And so we're looking at those in the future. We really want to be able to have increased resolution of neuronal subtypes. The goal here is to be able to discriminate between Alzheimer's disease and other forms of dementia. And so that is definitely something that's on our radar that we want to do moving forward. And all of these are possible because of this kind of expanded capability that is made available with the ONT platform. I'll show you right here the way that we think about this process. What I've just shown you is something that we've done internally for our own assay, but we're also working with other groups to help tailor this technology for what they need, either in a diagnostic test that they're hoping to build or in just some very specific research questions. So our pipeline is that we use what we call direct whole methylome sequencing. This is using the Oxford Nanopore platform for a biomarker discovery. This can be done at even low pass, 5x to 10x sequencing. We then have built our own analytical pipeline called the Methyl-Seeker package. This enables us to take that large amounts of data that we get from ONT and analyze it in a fast and simple way. We generate our models using this, and then we have methods that enable us to perform targeted methylation sequencing that we've developed over the past few years that are done without any amplification and without any bisulfite conversion. And this allows us to, if we're only worried about a very small number of targets, we can perform that targeted sequencing to really reduce cost and enable us to scale. At that point, then we start working on clinical deployment and kind of fit for purpose clinical validation. We can then scale this up. We currently have a 20,000 square foot laboratory here in Utah that enables us to do this. So we're really excited about where we are at this point, and very, very excited about utilizing the Oxford Nanopore platform for what we're doing. It provides, again, very unique capabilities in that it enables us to look at just the native DNA, how it comes to us. It's size agnostic. We're not worried about having to chop things up to perform our analysis, and it doesn't require any sort of amplification or modification to the DNA for us to be able to see the signatures that we care the most about. So really grateful for, again, the opportunity to visit with you all in this context, and excited to hand this over to Dr. Jeff Burns to add a little bit more insight.
All right, great. Well, it's great to be here. Thank you. Yeah, so my goal right now is to do a quick tour of the state of the field, the state of the Alzheimer's field, really to give you an idea of what this new technology is sort of moving into. And I'm really excited about this new technology because I think, as you'll see, there's a lot of gaps to fill. And sort of direct measures of neurodegeneration could really be game changers. So So and also it really is a new era for brain health. There's been a huge amount of change and transformation. So I'll give you a sense of what's going on. First, my disclosures. But really over the last 20, I've been at it for probably 24 years, and probably for the first 20 years, I had the exact same conversation with every patient I had about diagnosis, about the drugs, what was available and how we did things. And really in the last probably 2 years, there's been a dramatic transformation in terms of what we're doing in the clinic with our patients. And that's come about because of two things. One, we now have blood tests that allow us to assess the likelihood of amyloid protein buildup in the brain. And we have new drugs that go after that amyloid protein. And these new drugs are so basically we have a sort of mutually reinforcing need for one and the other. The new drugs need biomarkers to choose the right patients, and but it's a whole new world in terms of how we make a diagnosis and how we think about treatment with these new advances. And so that's really driving an increasing need and increasing movement and advances in the field. But I'm going to take a really quick step back and just say, all right, what is dementia? So dementia is defined by these things: a decline in cognition that interferes with everyday function. So when I'm sitting with a patient and their family, I'm asking questions about this. And there's no real magic to it. We're learning about what's going on day to day and how long those things have been going on, what's changed, and does it interfere with daily function? And then we think, all right, when we see that, what's the cause? And dementia is an umbrella term, and there's a lot of causes. The number one is Alzheimer's disease. But there's others. There's things like Lewy body related pathology, dementia with Lewy bodies, Parkinson's disease are associated with dementia, stroke-related dementia, frontotemporal dementias, which are related to ALS in some way. So there's a lot of things that we're thinking about. And what's the definition of Alzheimer's? Well, the current definition of Alzheimer's is the presence of amyloid plaques and neurofibrillary tangles in the brain. And up until really just a few years ago, we had to wait until somebody died to look at the brain under the microscope to know if they had amyloid plaques and tangles. And so our diagnosis of Alzheimer's disease is largely one where we're taking an educated guess as to whether or not this person has the presence of amyloid plaques and tangles. Now, with the advances in biomarkers, especially blood biomarkers, we're really moving quickly into this new age of confirmation of the presence of amyloid plaques and tangles in the brain. And so just again, what's the difference between dementia and Alzheimer's disease? The number one question I get almost every day. Well, they both have they're the same syndrome of dementia, memory and thinking problems that interfere with daily function. Dementia has many causes. Alzheimer's, the quote cause, is amyloid plaques and tangles, the buildup of these plaques and tangles. Now, over the last decade, we've really been improving our vision into the brain, really driven over the last 20 years by advances in molecular imaging, where we're able to inject a radio label into the bloodstream. It crosses the blood-brain barrier, and that label is designed to stick to amyloid or tau, emit a signal, little radioactive tip that the PET scan picks up. And in 2004, 2005, we began to see in real life, in living individuals, the presence or absence of these molecular pathologies. And that is what has really driven over the last decade major advances in blood tests to detect Alzheimer's changes. So where we are today is we now have our very first FDA-approved measures that allow us to detect the likelihood of the presence of amyloid plaques in the brain. And we're using this. We're using it every day. In the last year, our health system, we've adopted it. We've introduced it into the health system. We've had about 1,400 blood tests ordered in 6 months. And these measures are really important in terms of selecting individuals for treatment, moving them down that treatment path, anti-amyloid therapies, but also in supporting a diagnosis. And we have ongoing research that's really moving quickly in terms of assessing tau tangles, inflammation, neural injury, alpha-synuclein, vascular measures. But what we don't have is those direct measures of neurodegeneration. Probably the closest thing is NFL measures, neurofilament light. which does reflect on some level neural injury in a very nonspecific way. But where are we in terms of understanding Alzheimer's? That greater vision into the brain is really illuminating the fact that Alzheimer's has a lot of complex things going on. It's not as simple as the presence of amyloid in the brain. In fact, many individuals, probably 25% of those over the age of 65, have amyloid in the brain and they don't have memory problems. They may not have neurodegeneration. 25% have amyloid in the brain and don't have evidence of memory loss. But that is the primary measure we're using right now to make a diagnosis of Alzheimer's disease. And so amyloid's associated with neurodegeneration, but is not necessarily causal. We're still working on that, but tau tangles are associated and much more tightly linked to clinical decline. Lewy bodies are often present in people we call Alzheimer's disease. If we look at the brain under the microscope, we see Lewy bodies in probably 30%. And another protein called TDP-43 is very commonly seen in the brain in people with Alzheimer's disease. So it's a much more complex disease than simply amyloid and even simply amyloid and tau. And we also have vascular changes that are usually contributing. And so really the future diagnostics need to help us understand the overlapping nature of these diseases and really get at the neurodegeneration. These protein measures are good and very valuable today, but what we really need is to understand how are they overlapping, and then how are they influencing more directly these measures of neurodegeneration. This is what the future will bring, is really, I believe, a better sort of nomenclature for understanding how these things overlap and co-occur, but really getting at, why do they overlap and co-occur, and then, how are they producing neurodegeneration? And so looking ahead, I think, diagnostics and treatment, we're moving quickly, and I believe, and I'm very optimistic, that these new measures of neurodegeneration, this new technology is really going to help us understand, really be a game changer in terms of understanding how all these things interact, and also be a more direct measure of neurodegeneration. But what we need are more blood measures of the various pathologies. We need those more direct measures of neurodegeneration. We need to add in things like lifestyle and risk scores into our ability to assess a patient on a personalized level and look to the future, and then go after those changes and attack it from all angles. Right now, we're attacking Alzheimer's from the amyloid level. It's not a cure. It does slow things down. But I think that makes the case that, we can't expect a single magic bullet. We're going to have to look at a variety of things for individuals and come up sort of with a cocktail approach where we're attacking Alzheimer's in an individualized way from a lot of different directions. And so that's the state of the field that this new technology is stepping into. And I hope you can see that there's a lot of gaps, and that there's a lot of promise, I think, from these technologies, and a lot of opportunity to really illuminate, what's truly happening. Measure the neurodegeneration, maybe detect it early, maybe that 10% of those cognitively normal individuals where we're seeing, some evidence of neurodegeneration are the ones we need to be targeting for prevention therapies, et cetera. So a lot of opportunity, and I think I have a lot of optimism that these tools are really going to help change the game and continue to advance us into the future, where we're able to better treat, but more importantly, prevent Alzheimer's disease. So thank you.